Welcome back, grade 12s. We're here to finish off chapter 17. We're going to cover gametogenesis, we're going to cover life cycles, and we're going to cover non-disjunction or abnormal meiosis. So let's get right to it. All right, gametogenesis, forming of the gametes, these sex cells, because of course remember that meiosis, that's the whole purpose, is to form gametes in order to form sex cells. Sex cells are the sperm and egg in humans, but the sex cells or gametes can be different depending on what kind of species we're talking about. In the case of some plants, it's going to be spores that are formed. The two forms of gametogenesis for humans, of course, are spermatogenesis, the forming of the sperm, and oogenesis, the production of egg cells. We're going to compare these two processes. There's three major differences that we're going to talk about very quickly: the number of cells that are produced, the rate of gamete production, and the timing of the gamete development. So getting into number one, the number of sex cells produced. We know that with spermatogenesis, we're going to get 4 sperm cells when meiosis is done, which is typical, right? 4 haploid cells for meiosis. However, when it comes to the formation of an ovum, we're only going to get 1 functioning ovum. We actually end up with these 3 polar bodies, which are non-functional. And the reason for this is that during the cytokinesis events, only 1 cell, 1 daughter cell, gets the majority of the cytoplasm. And so that smaller cell, which we call the polar body, is not going to be functional. And that's why the egg cell is so much larger than individual sperm. So we can see that right here on this summary slide, 4 versus 1. So that's the number difference. Okay, we don't have to say too much about these polar bodies. That's not too curricular to have to know too much about them. But they don't survive because of the lack of cytoplasm. Now, moving on to number 2, the second difference is gamete production. We know that males are producing sperm cells daily, hundreds of thousands of them, and during our whole reproductive lifespan. Whereas females produce all of their oocyte cells during embryonic development, and number 3, if we look at the timing of gamete development, we know that for males it's continuous, ongoing development of sperm throughout our reproductive life. For the females, at puberty, when there's approximately 400,000 primary follicles within the ovary, they end up in arrested development in the womb in an early stage of meiosis 1. And so during the monthly cycle, that's when we pick up with meiosis 1 and finish it off for those growing follicles. And then we know that meiosis 2, there's that second division which occurs in the oviduct, the fallopian tube. Now, we're going to turn our attention to talking about life cycles. Now, we've seen the human life cycle before. There it is. Now, guys, in these life cycle diagrams, which you will definitely have on future tests and quizzes, you have to determine when things are haploid, remember, half the number of chromosomes, when things are diploid, and the names of processes like identifying where mitosis is happening or where meiosis is happening. So if we follow this life cycle very quickly, let's start here with our adults. So the adults, each adult goes through the process of meiosis to create the sperm. So adults are diploid. We use that symbol 2N. So they have the full complement of chromosomes in their cells, which for humans is 46. But going through the process of meiosis, we know that the gametes, the egg and sperm, we're going to cut that chromosome number in half. So now we have N number of chromosomes, which of course would be 23. Then if we follow the life cycle, we know that sperm can meet egg in the process of fertilization, and we form our single cell zygote, Well, when two sex cells or gametes come together, we know that we have gone from N to 2N when they're together to form the zygote. Then whenever we see growth in a life cycle, we can presume that mitosis is occurring, because that's one of the processes of mitosis, is differentiation and growth. So we know that since the zygote is 2N, simple growth continues to keep the same number of chromosomes, so the juvenile or baby is 2N, and then of course we just get more mitosis occurring. And so the human life cycle is pretty straightforward, but you're going to be seeing life cycles that you're not as familiar with. All right, especially when you see ones like plants, for example. So let's take a look at some more abnormal life cycles in just a minute. So before we get there, let's just go back to our chromosomes for a minute. Remember that we have 46 chromosomes, but only 2 of them are the sex chromosomes. Most of you are familiar with the fact that there's the X and the Y chromosome. Female is going to have 2 X's, but a male is going to have 1 X and 1 Y. All the other chromosomes, so this is good vocabulary to know, all the other chromosomes are called autosomal chromosomes, apart from the sex chromosomes. The X chromosome has most of the genes of the sex chromosomes, of course, because females don't have a Y. So there really is only one significant gene on the Y chromosome, and that's to make males male. So there again, there we're talking about males have the 22 autosomal pairs, an X and a Y. Females have the 22 pairs, but they have an XX, which would be a homologous chromosomes. Okay. Really, the sex of an offspring could be argued, of course, that it's the sperm that determines the sex of the offspring, because it's either carrying the X or the Y, whereas all eggs or ova simply carry an X, because that's all that a female has to donate genetically. The male can donate an X or a Y. So in the case of Henry VIII here, who had wives simply beheaded or killed because perhaps they couldn't provide an heir, a male heir to the throne, he really should have been looking in the mirror, because it essentially was his problem, because his Y sperm didn't seem to be able to find the egg. Okay, so back to our life cycles. We already talked about a human life cycle. Now we don't have the actual diagrams of babies or the adults. We just have words really representing what's going on. But everything is kind of where it was in that earlier diagram. We have our adults. We go through meiosis to create haploid cells, the gametes. Then when they come together, we create diploid cells. So in a human life cycle, it's like you can draw a line through the life cycle and have a diploid side and a haploid side. That's very common for most life cycles. So if we look at the lodgepole pine life cycle, we can see some different terminology vocabulary. So let's start with our adult organism, which in this case would be a mature tree. Now the mature tree is called a sporophyte. So a biological term there. So again, we're diploid. When you have the mature organism, we're diploid. Then through the process of meiosis, we create spores. So the spores are the gametes. And of course now, through a process of meiosis, we have something smaller, and it's haploid, which would just be N. But here's a big difference with something like this plant. The spores actually go through some growth and development, which is mitosis. They get larger. They become these things called gametophytes. But the gametophytes are still haploid, and it's the gametophytes that produce the gametes. And then the gametes, of course, can fuse at fertilization to become a zygote, familiar term, but now we're in the diploid part of the life cycle. And then to end this life cycle, the zygote will go through mitosis to become the mature tree. And that's a fairly typical plant life cycle. We have an adult. This time, if we find our adult, remember the sporophyte is called the adult. So this time, don't always get used to diploid being on top, haploid being on the bottom, because this time, if we cut this life cycle in half, the diploid generation is on the right. There's our mature plant. We go through meiosis, we create these spores. The spores develop and grow by mitosis to form a gametophyte. Gametophyte, that's indicative that it's haploid, 1N. We go through mitosis to create some form of gamete, which comes together at fertilization to become part of this diploid side of things. I think we have one more life cycle, at least we can look at here, all right? So we have our adult plant here. We have a female part of a flower. Remember that flowers are hermaphroditic. They are both male and female in their parts. We end up with forming pollen grains on the male side. That would be a meiosis process, okay? And then the meiosis process on the female side, we form an ovule, right? And then when pollen finds ovule, then we get our growing embryo, which would become diploid. So again, here we're haploid, and we continue to be haploid even if the ovule goes through some development, the eggs go through some development, or the pollen goes through some development. All right? Okay. Here again, guys, we have our fern life cycle. So let's see if we can figure out where we would divide this in half. Let's start with our adult. So if we find our mature plant, here's our mature plant. We know this is the diploid. And then we go through a process of meiosis. So now we've entered the haploid half. We create a spore. The spore goes through mitosis, but we're still in the haploid side of this life cycle, and it becomes a gametophyte. And then the gametophyte eventually creates structures. Remember, it's hermaphroditic, both male and female. We end up with egg and sperm in this diagram. So this is the process of meiosis right here. And then they can combine to form our zygote. So now we're diploid. So we would kind of be cutting it in half, right? Kind of at a diagonal downward cutting in half here, between this side all being haploid and this side all being diploid. Notice the zygote goes through to mitosis right away to create the beginnings of the mature plant. Now, last thing to talk about, guys, is abnormal meiosis. What happens if meiosis goes wrong? Well, that's going to involve this vocabulary term called non-disjunction. So what is non-disjunction? It's when we don't have a normal segregation or separation of the homologous chromosomes, which would be in meiosis 1, or the chromatids in meiosis 2. So if you remember what phase that most likely it would mean is affected, that's going to be the anaphase of either anaphase 1 or anaphase 2. Now, what's the product of non-disjunction? You can get a gamete with both members of a homologous pair, or you can get a gamete with no members representative of that pair. So you end up with one extra chromosome in your gametes. So in humans, that would be a 24 chromosome sperm, for example, or one less, which would be 22. And yes, it could occur in mitosis as well, but why would it be more serious if created in meiosis? Well, remember that in meiosis, you're creating sex cells. And so when the sex cells form the zygote, and you get that one cell, and that cell goes through mitosis, that abnormality would affect every single cell in that organism as it grows by mitosis. Whereas if it just happens in mitosis, say for example, in some skin cells that go through mitosis, only a very small number of cells of that organism would be affected, as opposed to the whole organism. So if we see this happening, for example, this would be our metaphase 1, and then our anaphase 1, we can see that the result of at the end of meiosis 2 would create 2 gametes with an extra chromosome and 2 gametes with one less chromosome. So again, we see this playing out in these diagrams here. And so that if you had a normal sperm cell meeting an egg cell where non-disjunction had occurred, so that we actually have 3 copies, okay, or an extra chromosome, then we end up with a zygote that has an extra chromosome. Now that creates too much genetic information or too little genetic information, both of which can cause significant problems in the embryo or the organism. And so we end up with these vocabulary terms. If we have an extra chromosome, that's going to be a trisomy event. If we have one less chromosome, we end up with what's called a monosomy event. And the most common trisomy is trisomy 21, which means all your chromosomes are numbered. So number 21 chromosome, if you end up with an extra copy, so instead of having the 2 you're supposed to, you end up with 3, then you have a situation of Down syndrome. And not that you have to memorize too many of the symptoms of the disorder, but many of us are familiar with someone that might have Down syndrome. So often mental challenges, short in height, enlarged tongue, maybe difficulty speaking, broad flat face, so there's some predominant facial features. Your life expectancy may be considerably reduced, and the incidence is 1 in about 600 births, and it does increase with older mums. Other autosomal non-disjunction disorders, trisomy 18, so having an extra 18th chromosome, or trisomy 13, an extra 13 chromosome. Now these problems are quite significant, and usually an individual might not even be born or not live very long shortly after birth, because these are significant chromosomal problems, all right? The life expectancy may only be weeks. If there are other autosomal non-disjunctions, so say trisomy 16 or trisomy 2, those are even more significantly challenged genetically, and the baby wouldn't even go to term. You'd have some kind of spontaneous abortion or miscarriage event. Now there are some sex chromosome non-disjunction events. So for example, you could have Turner syndrome, which is a situation where the individual only has an X. So they don't receive another chromosome, another sex chromosome. And of course, this individual would be female, fairly rare, 1 in every 6,000 births or 1 in every 3,000 female babies, all right? And an individual with Turner's, I don't have time to get into all the symptoms, but they will most likely not develop sexually. And they might actually have the appearance of a girl for the majority of their adult life. Now, Klinefelter syndrome is a situation, it's trisomic, and we get an extra X chromosome. So this would be a male because of the Y chromosome. And again, 1 in about 1,000 births. All right, and some people would say that extra X causes maybe feminization of features. We can have XYY, again, that's a trisomy disorder, the extra Y chromosome. Some people might say, well, this person would be like super male since Y makes you male. And in some cases, that would mean extra testosterone, and extra testosterone causes things like aggression, muscle mass, being able to put on maybe more easily than without it. All right, and could we have super females, trisomy X? Well, yes. All right. Now, how can we determine if somebody has a trisomy or monosomy? Well, we can get a picture of their chromosomes arranged in those homologous pairs. So that is called a karyotype, and it's a technology you should be familiar with. So it could look something like this. Here's all the chromosomes, but we can kind of go through and find the homologous pairs. Notice how the chromosomes are numbered all the way up to 22 pairs based on their size and shape. Remember that the genes are going to be found commonly on the homologous chromosomes, and then there in this corner is the sex chromosomes, which we can determine the gender of this individual. So this is going to be a male, since we see the Y paired up with the X. All right, so here's some typical human karyotypes. We know that this is the female because, again, we see our 22 autosomes, autosomal pairs, and there's the one sex chromosome pair. It's the same, 2 X's, female. Here is our male because instead of another X, we see the Y. And then we should be able to identify disorders. Whoops. So if we looked at this chromosome, and I don't want to take too much time, I'll just point it out very quickly. We can see that there's 3 number 21 chromosomes, so that's a trisomy 21, and if you remember, that's a Down syndrome. Is it a male or a female? Well, there's the X, and this is a very large chromosome that resembles, so this would be female. Now, in the next few slides, there are kind of testing if you can determine what the issue is. So I'm just going to let you have a look at it, and I think the answer pops up on the screen. So I'll let some time go by before I let the answer come up. You've had enough time, hopefully, with this one. Okay, normal male. All right, like I said, I think, guys, I'm gonna just skip very quickly through quite a few examples here. You can have a quick look for those through the slideshow yourself. Last thing to talk about, guys, polyploidy. Now, polyploidy is defined as when an organism has more than those 2 complete sets of chromosomes. Remember that we have homologous pairs, one from mom and one from dad. But it is possible to have more than the 2 copies. You might have 3 copies or 4 copies. But remember, it's complete sets, not just one extra chromosome, as we were talking about before. Certain terms we use and symbols. Remember that haploid, and we've seen this already, is represented by N in humans, 23. Diploid represented by 2N, so most of our somatic cells or body cells are 2N, so that's 46. Triploidy would be having 3 sets of all the chromosomes, and tetraploidy 4 sets. Now in humans, very unlikely, not very common at all, but in some organisms, especially plants, there's huge numbers of organisms that have this polyploid situation. So it would look something like this. Now, how does it actually come about that you would become polyploid? Well, it's abnormal meiosis, but it's not just one chromosome that's left behind or an extra chromosome. It's actually failure of meiosis so that none of the chromosomes are split. All right, so we could have a diploid parent species here with 6 chromosomes, and if there's failure of meiosis completely, then in the gametes we'd end up with all those chromosomes. And if that happened twice, and then they actually got together, right, fertilized, we could end up with tetraploid situation, a zygote with 4 copies of all those chromosomes, all right? And the offspring may be viable and be self-fertile. So it can happen naturally, and it does happen with plants quite a bit. Quite often, sometimes the polyploid individual is not fertile, especially triploides, where you only have 3 copies. All right? We're not going to get into this fairly complicated situation here about creating, again, tetraploids from a diploid organism, but it's a little more complicated than we really need. All right? So there you go. That finishes chapter 17. Happy studying.